translation. We all know that in order for our bodies to function properly, it must be supplied with certain nutrients and important nutrients by eating food. Alright? Now, and your food cannot enter your bloodstream as it is and reach the cell as it is. They should be digested by chemical reactions inside your digestive system. And these chemical reactions need proteins and enzymes to happen. Okay, these proteins
enzymes to break down the
these proteins are
during
mainly
transcription.
These proteins are mainly instructed from the DNA structure. Okay, DNA is the genetic material that have genes which code for protein synthesis. Okay, so these proteins are synthesized according to the need of the body or the cells. All right, for this process to happen, first the DNA should be transcribed to mRNA molecule, as you see here.
The DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination.
The transcription, as we learned before, happen when one strand of DNA used as template for mRNA strand, okay, and this happens by the help of the RNA polymerase enzyme by attaching to the starting point until the whole mRNA formed, and this mRNA will have the message of the genes.
During initiation, the promoter region of the gene functions as a recognition site for RNA to open. Then during elongation, the RNA polymerase slides along the
as you see here, during transcription,
links
nucleotides of RNA are bind to one strand of DNA nucleotides. When in this part, the RNA will get the message from the nucleotides on certain gene on the DNA strand, and this message will be translated later for the protein needed by the body.
To the 3' end of the transcript is complete.
After the transcription complete, this DNA, this RNA will be this attached to from the enzyme and from the strand of the DNA, and they will leave out from the nucleus to the cytoplasm.
The messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription in this process is called RNA strand that can leave the
now
nucleus.
This RNA strand is mature. It can leave the nucleus. It goes out from the nucleus to the cytoplasm, as you see,
through a nuclear pore and enter the cytoplasm to begin translation.
Now this mRNA will begin translation, which means it will convert the message that coded in these nucleotides into a protein synthesis.
How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop.
Translation process should have a starting region and ending region. Okay, there is a codon for starting, which is start codon, and the end codon where the translation will stop when it finish. Okay, now as we said, each codon contains 3 letters, and these letters are the nucleotides. Okay,
begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA.
So, translation needs three parts. The first part is the ribosome, and the second part is the tRNA. tRNA is the transfer RNA, which transfer amino acids from the cytoplasm to the mRNA strand, okay? And ribosome has two parts, small part and the large subunit. You will see it now.
Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete.
Now, the translation will start. After recognizing the starting point here, the large subunit of the enzyme will come and attach to the mRNA, and now the translation will start, okay? The main function of the ribosome is to bind the amino acids together so the polypeptide chain forms.
In the large ribosomal subunit, there are three distinct regions called the E, P, and A sites. During elongation, a base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid.
Okay, so each anticodon in the tRNA corresponds for a particular amino acid. Okay?
A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer.
So here comes the function of the ribosome. The ribosome will bind the first amino acid to the second amino acid, and this tRNA will be uncharged, doesn't have any amino acid anymore, so they will leave the strand.
Transfer RNA molecule at the P site. The complex slides down one codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached.
And in this way, the amino acids are connected to each other, binding in a polypeptide chain, which is a protein. All right?
A release factor binds to the A site at a stop codon, and the polypeptide is...
At stop codon, this translation will stop, and the protein will be released
from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation.
It can begin the process again on another side of the